So we have another infectious disease outbreak on our hands. This time it's Zika. Not long ago, it was Ebola, and before that, it was SARS. And when we have these epidemics happen, the idea is that we send our scientists to the lab, and we ask them to go to the lab and find an immediate cure or a drug therapy. And the reality is, it isn't really like that. That's not how science works. Those investigators that are trying to find that quick cure are really working on a foundation of knowledge from basic research. So I'm going to tell you a story about my own research, which is on tuberculosis. But before I do that, I want to define to you what basic research is. So it's really scientific discovery for the sake of discovery. It's not about immediately thinking about making a cure or a new drug or a new vaccine, but it's going to the lab and discovering new things that happen. And at some point in the future, they may have application to actually curing a disease. So let me give you a couple of examples. So Richard Prum is an investigator in Yale. He's a really well-respected ornithologist that studies, he's really driven the concept that birds evolved from dinosaurs. But one of the projects in his lab is actually to study duck sex. And it seems at face value that that doesn't have a lot of scientific application about human health. And it has indeed received a lot of attention from our government and the public about whether they should have received funding. But I would argue that there is merit to this study as a basic scientist. So ducks, like humans, pair bond for life. And ducks, like humans, take part in coercive, violent sexual encounters, i.e. rape. So now you can see how there might be application to studying duck society, instead of taking that precarious path of asking those questions in the human population. So it may have relevance to human health and behavior. Christian Nieuwsland Volhard's lab studies fruit flies and development of fruit flies. And again, at face value, you might ask why we care about how fruit flies develop. They're those annoying things that are in our offices and in our homes. But it turns out that many of the genes that are related to development in a fruit fly are actually very similar to what we see in human embryos. So again, you can see how there's a translation of basic science into human health, and particularly in a topic that would be very difficult to answer in humans. But independent of Christian's discovery about embryogenesis, she actually made a really significant discovery, essentially accidentally, where she took out a gene in a fruit fly that we knew was important for development, and those fruit flies became sick. And they became sick from fungal or bacterial infections, and that's a picture here that you'll see. So what she discovered, serendipitously, was a series of receptors that are also present in humans that drive immune responses. And her discovery has now led to really smart and effective ways for us to develop new vaccines for human disease. Okay? So this was basic science that now has translational application to human health. So let me bring this closer to what I work on. I study the disease tuberculosis. It's a debilitating human disease. It kills 3 million people every single year. And we need effective vaccines for this infection, and that's really what my research is about. But we also need new drugs to help eradicate this infection from the population. And that's been extremely challenging to do. So when an individual is diagnosed with tuberculosis, they receive a cocktail of drugs, a 4-drug cocktail, a big handful of drugs. And they don't just take that drug for a week, like most of us would with our antibiotics. They actually take those drugs in combination for up to 6 to 9 months of duration. And so they're very toxic. They have really unpleasant side effects. And so many of those patients stop taking their drugs early. And that leads to redevelopment of tuberculosis, and they're sick, and they can transmit to others and infect other people. And it also means that there's a development of drug resistance in the population, which we know already is really detrimental to human health. So we need improved drugs, and we need drugs that work quicker. But despite the fact that we've been looking for new drugs for tuberculosis for decades, we haven't found a single drug that can kill the bacteria that causes tuberculosis in less than 3 months. I would personally state that I think this is because we're going about this the wrong way. We're thinking about drug discovery for tuberculosis in really the wrong way. So what we really want when we think about curing tuberculosis is the equivalent of a Z-Pak. We want a patient to go home with a 1-week therapy, and within 1 week they're cured and they're no longer infectious to others. But we haven't managed to achieve that. So this is how we feed the pipeline of drug discovery for tuberculosis. It's a very standard procedure. Scientists develop a panel of drugs, and then they test those drugs against the bacteria in a broth. And if some of those bacteria die, then those drugs will move forward to testing whether the drugs will kill the bacteria within a cell in a culture dish. And if some of those drugs work, they then move forward to test those in a small animal model, such as a mouse. And again, if some of those drugs are successful, then they move forward into higher animals, such as guinea pigs or monkeys, before they would ever go near a person. So along that pipeline, there isn't very much that resembles a human. So perhaps we're going about this in the wrong way. So how does my research fit into this? I'm an immunologist. I'm interested in how our immune system functions. I don't have a strong desire to develop new drugs, but I have an interest in curing tuberculosis. So as part of my research studying immune responses against tuberculosis, my lab made a new mouse. So we developed a mouse that was lacking a molecule called interleukin 10. And interleukin 10 is an interesting molecule because its job is to turn off immune responses. Whenever we generate an immune response, we also have to turn it off so we don't actually damage our own bodies. And interleukin 10 is the off signal that does that. So we deleted interleukin 10 because we believed it was important in driving tuberculosis disease in people. So when we infected mice with the bacteria that causes tuberculosis, we actually found that in our mice that didn't have interleukin 10, they did much better. They were able to control the infection much better, which told us that interleukin 10 was actually driving disease in our animals, and we believe that that also happens in humans. This wasn't a surprising finding for us. This was actually validation from many years of research that it implicated this molecule in susceptibility to have tuberculosis. And so that's not really the purpose of this talk. While we did this research purposefully to understand IL-10, we actually made other discoveries that were relevant to drug development for tuberculosis. And the first discovery that we made, an entirely unexpected discovery for us, was that when we looked in the lungs of those mice that we'd infected, they developed what we call granulomas, cellular structures in the lung. And those granulomas looked exactly like we see in humans. So mice don't make granulomas that look like humans, and yet we test drugs in them. But our mouse developed a granuloma that looked just like a granuloma in a human patient with tuberculosis. So this was a really interesting discovery and a really important discovery because it's now given us a model that is better to model human disease. But that's not the only part of this story. Because we had a model that looked like a human, we obviously asked the question of whether it might be used to screen drugs for tuberculosis and perhaps be a more effective model to screen drugs for tuberculosis. And so we gave our mice a TB drug. And interestingly, what we found when we gave our mice a TB drug was that the drugs actually worked faster. So our mice that didn't have interleukin 10 cured much quicker than a mouse that had interleukin 10. So this was telling us something interesting about interleukin 10. It's inhibiting the way drugs are working. But we actually needed to ask a second question. We didn't know at this point whether that early cure was because of these really nice granuloma structures that our mice had that maybe made the drug work well, or whether it was really about interleukin 10 inhibiting our bodies and the function of the drug in our bodies. So we set up another experiment where we infected regular mice that we normally screen drugs in, and we treated them with TB drugs. And at the same time as that drug treatment, we actually blocked the activity of interleukin 10. And we asked whether that could accelerate the cure. And much to our surprise, we found that that's what happened. So the presence of interleukin 10 prevents the TB drugs working properly. What's important about this finding is that this would never have been discovered using our standard drug screening methods. So the individuals doing drug screening didn't think about how the host was interacting with the drugs and could stop the drugs working effectively. So now we know that we can take out and modify some host molecules, okay, molecules in our own bodies, and we can make those drugs work faster. And that means that our drug developers can now take that information and go and make drugs that work much faster against TB. So we're not at the place where we can send somebody home with a Z-Pack, but we have been able, with one simple modification, to shrink drug therapy down from 6 months to 6 weeks. So this is a huge gain for patients that have tuberculosis. And it was really, we came about this from doing basic research in the lab with no actual goal of developing a drug, but that information is now relevant to the development of good drugs for tuberculosis. So I hope from this that you take home that basic research is important. Discovery for the sake of discovery has a lot of value for human health. And I hope you also take home from this that when you think about where the money should go, it's obvious that we should try and cure some of these outbreaks. But there's also a lot of merit in putting money into basic science, because that's the pipeline that feeds the cures for these diseases in the future.